So the topic which I am going to discuss little bit more on light arresting, degradation of light arresting complexes. Degradation of LHCs during acclimation and senescence. Maybe a little bit in the inside will be little bit different, but mostly we'll be talking about light harvesting complexes. LHCs means light harvesting complexes. So this might have discussed with the HRO, but I also told you in the last first class, the light arresting complexes here, this is from plants and this from the lower organism, cyanobacteria. So this is the cyanobacterial thylakoid schematic representation, and this is the schematic representation of higher plants and algae photosynthetic complexes. So since we are talking about light arresting complexes, how they arrange, just we'll discuss. So this membrane complex, thylakoid membrane, you see that the linear structure where comprises with the lipids. You can see the bilayer, lipid bilayer, this one. Then you have we also discussed last time, now we have a core complexes that's called reaction center, and they have a D1, D2, CP43, 40. Or algae or any photosynthetic organisms that would be harvested by light arresting complexes. Light will be absorbed by light arresting complexes. So these and transfer that the light, to the photons to the reaction center where water get split. Now whatever the water, and you get a oxygen and protons, and we discussed last class also the hydrogen energy will be coming through this photolysis of water. So if it is coming to the cyanobacteria, so they have and phycocyanin, this one PC, and the reddish color, the one phycoerythrin. So all these pigments are present in cyanobacteria, and also they will be also present in all red algae. So last class when we discussed why Niagara is bluish color, why most of the lakes are very colorful, why is called Red Sea. So Red Sea is why it's called Red Sea. Any of you can tell? Huh? It will be having more blood. No? Huh? Anthocyanin and other thing, but mostly because of the phycoerythrin. So that's why it looks like a Red Sea because it comprises the bacteria, algal anax. Onto the water, whatever it, because of reflection you can see Red Sea, and Black Sea also there, Red Sea, Black Sea, whatever, most of them it will be bluish. That's why it's called cyanobacteria, blue green algae. Okay, so higher plants are algae. This is the core, similar to this. This is the core reaction center, and you have a surrounded by this, the surround, to the core. Similarly, you have photosystem 1. The number of light harvesting complexes varies from plant to plant, and also algal have a more light harvesting complex because they live in deep water or fresh or the sea water, where it require more light harvesting complex. That's why they have more light harvesting complexes. So if you look at this one, they say that these are the proteins and their respective genes. You represent in italic letter, then it will become a gene. Similarly, alpha-glycosanin, they may have several encoding genes. These are all, spicosanin, several encoding genes. Picorithin, there are many, several encoding. But light arresting complexes, this is about 5 in photosystem 1 and about 6 to 7 in photosystem 2. These are the light arresting complex about the structure. So now, little bit we'll go about physiology and other, the biochemical part. So now if you see this video, plants exposed to various kind of environmental conditions. So it was, mostly you see that, then when seasonal changes, it changes turn into from spring to, it will go to autumn, where you see a lot of changes on to the plant. Now they're green, now it will become yellowish color, we just discussed, like become anthocyanin and other carotenoid pigment they convert. And now this is a cycle. So I don't know, winter, and most of the, and the question comes here, maybe you can answer me. Any of you been to Himalayas? Yeah, and winter season. In winter season, and where it gets snow of this much height, and most of the European and US and Canada, they will have a snow full of snow. The snow, and you don't see any leaf. There won't be any leaf. There won't be any except pine trees. All trees doesn't have any leaves. The question is here whether they can perform photosynthesis. How they live? They know photosynthesis. How they get food? Except pine, all other plants, apple, you say that apple, mostly an apple, berries, all other, any other crop. So they don't perform photosynthesis because there is no green stuff there. Everything is gone, all leaf fall. So it looks like this, you see the snow and no leaves. Of course, anyway, so the water will be there in all, they will be alive. Once the season is over, spring comes, then it blooms and you get the photosynthesis. When even if the sun is there, in places like US and Canada in the winter season, they will be sunny, but cold is about minus 10 degrees, so they cannot live. One thing here is, there is a yeah, it won't be hibernation. Hibernation means they don't do, but they have a cyclic electron transport where they don't need any light, but they do circulate energy through metabolic pathway. Metabolism will be active. Through metabolism, you get the ATP. ATP will be generated. It will be a cycle. The electrons come degraded, Kelvin-Benson cycle or something by Maillard biosynthesis, where the electrons will pump into it, makes a plant continuously ATP generated, and plants will be alive. So plants will be alive, but photosynthesis happens, but not active linear electron transport. Okay, so now, so during that time, what happened? Plants, as I said, this is called you can also call acclimation, or all these process exposed, or in all these process will be can see in during the plants or algae exposed to different conditions. So here there is a called non-photochemical quenching. Non-photochemical quenching. So the meaning is, here you can see here. So let's take from here to here. That means seconds, minutes, hours, and days exposure of the plant or the algal system. There will be several things happen. The seconds, if you see the seconds, so now this is called short term, and long term is basically hours to days. So during that time, these are all the events can happen. Change in electron transport, formation of delta pH, that I will tell you a little bit later, and picky pull reduced state, and photoxis, and deepoxidation of xanthophyll cycle, and increased xanthophyll cycle. If you go into little bit more hours together, you can see up to short these will be there, and longer duration of the the plants, they will be adapted to several these mechanisms, transcriptional changes, protein degradation, and PS2 repair. That's what I'm going to discuss. The light arresting complexes will be get degraded. That was a change in the antenna and change in stoichiometry of PS2, PS1, and accumulation of a lot of antioxidant. This can happen. So the non-photochemical quenching is basically, again here you can see here what is non-photochemical quenching. So if you look at the thylakoid membrane, so thylakoid membrane, as I said, the linear, where you have a lamina under the peripheral and the lumen. Lumen is downside. When the electron is pumping from top to down, the protons, whatever is the protons which you talk about, hydrogen ions, the hydrogen ions will be pumped more towards the peripheral to the luminal side, and where that will become more acidic. The down will be more acidic. The pH will be acidic, and the acidic is the sense is a favor to the chemiosmosis. Chemiosmotic means again the production of ATP. So the chemiosmosis is a down state, down of the lumen. It goes down and goes to the ATP motor. So then where you may have many things happen, a lot of the cyclic electron transport will be more activated, but however the delta pH is nothing but a luminal pH become acidic. During that time, there will be a QE, that is the one, QZ is another one, and QT is other one, and QI is another. Three types of the four types of adaptation. So QE is nothing but this is called delta pH will be more increased. Through that there will be a specific genes or proteins, that is delta pH, LHCSR, or is also called PSBS, photosystem reaction center protein, that will be overexpressed by acidic pH, and yet this component will be non-photochemical quenching. There is another one, non-photochemical quenching. Here is the one, this is the one. There is a pigment called carotenoids. I said we talk about carotenoids. The plants, when it goes to any kind of stress, the plants is trying to convert the chlorophyll pigments to the carotenoid pigments, where they will be photoprotecting. Photo means when the light is more, they try to get protected by photoprotecting, where the zeaxanthin is a pigment, convert more to excess light will be quenched. Quench means reduce the light. So that means even if it is falling lot of light on the quantum of light, it try to take only the light which is required, and where the zeaxanthin pigment is the one, it controls the light. So that is the meaning of that. There is another one, this is called state transitions. There is one that we will be discussing later. There is a enzyme called STT kinase. That kinase getting phosphorylated, that is called QT is the one, that we will discuss bit later. And this another one, if it is, let's say, you can say that, cat in the room, you hit the cat, just you irritate cat, cat won't do anything. If you irritate cat, what it does? it will become pulley, it will become tiger, right? Cat will become tiger. So same thing here. So what it does, you keep stressing the plant too much, it get damaged. So that's what it's called photo inhibition. Even photo inhibition also, it can be, there will be some repair mechanism is there. Still, there will be trying to protect. That's what, if somebody is hitting you, that means, when you hit cat, it try to protect he or she himself, so that, there is something still, I will be alive. So this is the way, it's called, the photo inhibition. So these are the, the mechanisms the algae and plants can protect themselves. So there is a other mechanism, as said, redox regulated phosphorylation of light arresting complexes, which is the one called state transitions. So now you all know that by this time you might have learned what is PS2, what is photosystem 1, right? HRO has described. PS2 absorbs light at which wavelength? 680 nanometer. PS2 absorb light at 700 nanometer. Why so? Right? That also I discussed. But this difference, when you see the electron transport chain, the Z scheme, or you call it as a Z scheme, there is a voltage, significant voltage difference in there. This voltage difference from photosystem 2 to photosystem 1 is balanced by this so-called state transition mechanism. That's called light, the redox regulatory mechanism. So it will be like a motor, the transport system, it's going like a circle, which we're going to discuss here. What it does here. So now the photosystem linear electron transport, if you get photo 680 nanometer, excite the electrons and goes to the PQ pool, then PQ pool to the cytochrome B6F complex, then it goes to the photosystem 1, it will be like that cycle. So the PQ pool here, what it does, it will be, this is photosystem 2 light arresting complex, two of them. And let's take a photosystem 1. There is a photosystem 2 and photosystem 1, two different. So when the system required to balance the energy, so when it is now let's say if you're going outside, if you're going to the outside, plant exposed to fluctuating light conditions, there will be fluctuating light. That means rainy season, you get a hot. I mean, yesterday, day before yesterday, we could have seen it will be shiny, then again it get reduced. Or even if it is a gradually increased light, so what it does, the PQ pool get reduced. That means plastoquinone pool is oxidized reduced mechanism. That is redox regulated mechanism. It get oxidized and reduced. Oxidized, reduced. When it getting reduced, what it does, through LH2 kinase, it get phosphorylated. You see, ATP will become ADP. That means one of the phosphate group is the ADP, diphosphate. From triphosphate to diphosphate, the one of the phosphate group is going to attach here. That is this. It will become the light arresting kinase. What it does, the kinase of LHC2 and phosphate group is attached. This phosphate group is now is going to be here poised. That means they get together, and it will become a photosystem 2 here. Photosystem 1, I mean poised means with the photosystem, with the LHC2 is associated with always photosystem 2. When it get phosphorylated, this in general biochemistry, if anything, any phosphorylated form is stable or unstable. Any phosphorylated form is unstable. So unstable molecule cannot be, they'll be like right, if you are tensed, right, you will be unstable, your mood will be fluctuating. Similarly, here also, when it is getting reduced form never will be sit with the photosystem 2, it has to migrate to photosystem 1. That's what it does here. Photosystem 1 light arresting complexes, complex it form. So then what it does is called state true condition. That means when light is exposed by the plant, that means mostly blue light, that means visible light, solar region, this takes the light and get phosphorylated, then migrated to the photosystem, light arresting complex 2 will migrate to photosystem 1. Then then what it does, my job is done, right? The vehicle, let's take from here to Hyderabad to, let's take Rapido, right? You can send Rapido food delivery. So food delivery, deliver, and what it does, that guy will go empty. So empty, what it does, again you go to whatever Swiggy, whatever Zomato, you go to again shop Paradise Biryani, get Paradise Biryani delivered, again he has to go and deliver again. Similarly, what it does, it get oxidized, the picky pool get oxidized, and this using phosphatase enzyme. That is a kinase enzyme. Kinase enzyme is always ATP will be become ADP, phosphate group is attached. Now here phosphate enzyme, what it does, again ADP will become ATP. So the phosphate is removed and ATP will now be formed. Then all of them, these will be also dissociated. The light arresting complexes, they will be dissociated. Again, this is the cycle. This is called state 1 transition. Oxidized is state 1, reduced is state 2. So, and different light conditions. It requires different light conditions. Here is, it favors to photosystem 2, it favors to photosystem 1. Photosystem light 1 is longer wavelength and photosystem 2 is shorter wavelength. So this is called state transition mechanism. Through light arresting complex, it happens. I mean, little more to schematic, more representation. So this is the one, these are the kinases, STT7 in algae, STT7 is in plants, and this is reduced condition, and this is the oxidized condition. This is PPH1 and TAP38, these are the phosphate enzymes, which is involved in oxidation. You can see that light arresting complex is attached. So then it does, the photosystem 1 is going to attach here, where you can see here the light arresting complex 2 and light arresting complex 1, this phosphorylated form is attached. Then when it get oxidized, it will go, only light arresting complex 2. So this is the mechanism of state transitions. Okay. So we also showed this one from, this was one of my collaborated data, and also I also showed this one, my 20 years back story. When it is, this is the cell, you can say this is the chlamydomonas cell, this higher plant, Arabidopsis, sorry. So this is a cryo-electron microscope. Anything structure you want to see, cryo-electron microscope, you can, using that one, what we showed, how it is a regular, this entire light, whether, it's experimentally you have to show whether it is true or not. So you can see this is the photosystem 1. The phosphorylated form of light arresting complex is migrated. That's what, this data shows. So, just want to show a little bit more representation. So this is a PQ pool, get kinase is activated by kinase activates the LHC2 phosphorylation. So what it does now, so it get excited, it goes and bind to the photosystem 1. So this is called state transitions mechanism. Happens every plant, every photosynthetic organism. In phycobilisomes, if it even in cyanobacteria, phycobilisomes will be there. Even in anaxin bacteria, this system is there. So then what it does here, when it is migrated to, this is the one, you say that state 2, now it has to be migrated to state 1. So now what it does here, excited, and this goes and backs to dephosphorylation, it goes and binds the light arresting complex, go and migrate to the photosystem 2. So some of the data I would like to show here, but not much. From our data, from our laboratory, how light is induced state transitions influence the light arresting complexes. How different abiotic stress can induce like state transition that we wanted to understand, and what kind of signals are playing major role to inducing the PQ4. So the simple technique which low temperature is done today, tomorrow, yesterday, Monday. Okay. You will be showing this technique, fluorometry, simple fluorometry, where we show the difference between state 1 and state 2 complexes. That means we induce physically, that means light conditions, and then see that when photosystem 2 light arresting complexes, when it get excited, that means it get phosphorylated. The phosphorylated complex has to migrate to photosystem 1. That means, so let's take the balance. So if I have like this, so if I have put more balance here, so my weight will be, what happens? My weight will be increased. The weight get increased. So then what happens in the experiment, in simple terms, we need to show that one. The photosystem will be, it will become less, but we have normalized here. And you see that photosystem 1, this photosystem 2, and this is the photosystem 1. When light arresting complex is more, that means height is more, or the weight is more. You can see here, from here to here, there is a microscope. The junk of, we'll see here. So this is called major light arresting complexes, where to want to show this one biochemically, what we are showing here. So the LHC3, LHCb1, and LHCb2. That means 1, 2, 3, 3 subunit, 3 genes are very important for trimeric complex. This trimeric complex, which light arresting complex is very important, and which one is getting phosphorylated. That's what we wanted to show. So here, all of them are getting phosphorylated. B1, B2, B3, all of them are getting phosphorylated. We are matching here. So, but when you see that this is the antibody we used, antiphosphothrione. So then 2D electrophoresis, the technique is called one-dimensional electrophoresis and two-dimensional. It's called isoelectric focusing. We separate the proteins, then go with 2 dimensions on the SDS page. That means you can separate based on the molecular weight, where we showed here, again used antibody work and identified the LH. I don't want to go deep into the story. The LH2B is a major subunit or protein, is inducing state transitions. Of course, the other proteins are also inducing, but because of that, there is, you can see here, this is not state 1, 1, 2, 3. When you see here 1, 2, 3, 4, 1, 2, 3, 4, there is one of the isoforms getting complexes. That is an indication that LH2B play major role, really major role to induce the state transition. That's the meaning of that. Okay, then we separated complexes where it is getting associated with, to show more biochemical data. Sucrose density gradient, HRO, he has showed many things, and where we showed, the S2, they have an additional band that is called light arresting complex. Photosystem 2 to photosystem 1 is migrating. So let's take, if it is a dark condition, plants also exposed, let's say, talk about there is no light. What happens? That's what we wanted to prove. So can you understand what is it from this? From this? Huh? No, from these, I mean not the subject, I am not talking subject. From this, what do you expect? What do you can give a conclusion?
Person is the reason for everyone
No, simple.
support
These people are walking straightly, right? And these are all visually challenged people. They are also walking. That means in the dark cycle, in the dark, there is no light. These people also have to live the life. So they are also walking with the stick, with the support, they can walk, they are better than us. They know really, they also follow discipline. But this traffic, Indian traffic, so they could able to cross, I mean, I don't know whether it is Indians, but it looks like Indonesia. So they're able to cross the road. So they also can cross. I mean, there are bright people, they can see the light, they are also, but the meaning of the story here is, so even if it is stress, plants are, they become I mean, in the regular condition, that's called state condition. In the dark or in the fluctuating conditions or highlight something stress is there, they also plant can survive because of some mechanism that is also called state condition. That is called dark induced state condition. So whether it is true or not, we showed, we used Arabidopsis. So here we use a temperature. Just increase the temperature, can induce the state condition. They perfectly induce state condition. Then the mutant, they're not performing state condition. That means LHC2 kinase is still activated. The question, this is a lot of, to convince the reviewers, it was so difficult. These are all our elaborate data work. Convincing reviewers, this was a plant journal which we submitted. We are not able to convince them, but they asked us to do double mutant, but we could not be able to do it. It's Sridhar who was the first student who did all this work. Anyway, so but we got a lot of the convincing data. In the end, people have agreed that there is a dark induced state condition which we have discovered from our lab. So when any kind of stress also induce state conditions, but it may not be mimicking, but LHC2 is getting phosphorylated. That's what we wanted to show. This is the LHC2, they're nicely getting phosphorylated. But there are many other things happening. I don't want to give you more. I mean, this is the same thing. The phosphorylation is happening, and it's not happening in the mutant. Wild type it is happening, and mutant does not happening anything. And similarly, when you induce, you see that again stress, very nicely inducing, this individual LHC subunits of LHCs, they are getting phosphorylated, all of them. Okay, role of, I mean, I don't want to, this is a lot of biochemical data. We did a lot confirmative analysis. We used various inhibitors to show whether it is true or not, how they are getting. This is where we showed the really signal, what kind of signal is coming, where it is coming, the DCMU and all. They can inhibit the phosphorylation to show many things. I'm not going to discuss about this one. So this one also it is not required. Then we showed the migration, whether it is guide. So you can nicely shown that there is LH2 is getting, you see that 40 degrees nicely coming out. This is the purified complexes from sucrose density gradient. And we showed several other, the parameters to understand. This is a cyclic electron transport. This kind of thing is cyclic electron transport. So what we have connected, there is a linear electron cyclic electron transport. In the wild type, this is a linear. When it goes to the 40 degrees, when you incubate, I mean, cells or the leaves, when you incubate 40 degrees, they will induce state conditions, the state condition through cyclic electron. What we have showed from this data is, in regular light conditions, they don't induce state conditions. When it's a stress conditions or in a 40 degrees or anaerobic condition, or you induce any other stress condition, they induce cyclic electron transport, and in turn cyclic electron transport gives and activate light arresting complexes, then for the PQ pole, that is a plastoquinone pole, get reduced and light arresting complex are activated, then the electrons will be, then through that the phosphorylated light arresting 2 can migrate to the photosystem 1. We used, I don't want to show these are all the mutants, lot of mutants we use to show the migration. These are the two schematic representation. So what we said, through that there will be a cyclic electron transport in dark cycle, not light conditions. This is the cyclic, these routes are cyclic electron transport, PGR1 and PGR5 and NDH. The electrons are pumping through this one, and through this PQ pool is getting reduced. This one is the reduced, so that whatever you are getting, and we showed here linear teletransport and cyclic electron transport. So the photosystem 2 is getting reduced through the cyclic electron transport. That's what we wanted. I will skip here all this one. You don't need all this one. Yeah. So finally, what we showed, these are also a schema in algal system. So you will have a, we induced heat and show that the LHC2 getting phosphorylated and associated with photosystem 1. You can see here, this is what in an algal system also it is happening. So further, this will be a next Monday. I'm going to discuss the other thing, the chloroplast adaptation or chloroplast development in light condition. There is one more topic that I will discuss. So if you have any queries or clarification, I'll be happy to clarify your doubts. Yes. Yeah, that's very good question. So that's what it tells. Your spectrum, the solar spectrum, even an algae or plant, when you change different light conditions, the blue light is fine. Blue light is always good for the blue light. That's what when you light shows there is the WebGR color, but plant absorbs two lights, that is a blue and red light. These two are the major one. So blue light and red light combination, if you give it, it's good. If you give only blue light, so you are only favoring to photosystem 2. You are not favoring photosystem 1. So you have to give blue light, or even if you can give fluctuating, that's fine. They can grow nicely. So let's say if you give only red light, plants cannot grow. Only specific, let's say 700 nanometers, plants will be stressed. And if you have to give, that's why we give always solar, the 400 to 800 nanometers or 400 to 750 nanometers. Did I answer you? Yeah, exactly. Yeah, you are right. This is called again adaptation. So the pigments and algal or plants, now you need to have let's say a commercial importance. You can give, you are changing, something you are changing, you are getting something out of it. So you want to have a, let's say where Pavitra was she never observed any pigmentation. That means we are giving a right condition of environment. We want to have a, let's say a pigment, then we change the conditions, either light or nutrient. So light, you go with red light. What you give, if you give more of red light, mostly pigments will get changed. Chlorophylls will be changing. It has to be changed, chlorophyll, and it favors more of photosystem 1. So there will be, if it is more photosystem 1, you may have to have a lot of xanthophyll pigments will be accumulated. More xanthophyll pigments will be accumulated. Let's take if you give a blue light, more of blue light. So there will be a condition of lutein will be increased. Mostly lutein will be increased. Likewise, you can change for a commercial purpose. Yes, you have to change to get the pigments. You are not benefiting the algal system or plant system, but to human selfishness. No, you want to get something. Yes, you have to change something. The parameters you get different conditions. That is the same thing. Like there is Urban Kisan company. There is they do vegetables, ground roof vegetables, where they do all these changes. So what they say that the any amaranthus family or greeny leaves. So you get nice anthocyanin color, complete anthocyanin color. What I work for them as the consultant. So we change different light conditions, and they get all these like a beetroot color leaves. That is only because of we didn't manipulate any genetical manipulation. We manipulated light, got different color. So that is now in the market. You can see, have you seen any of you? The leaves, there are different color of leaves. That is only because of light. If you change something genes, yes, it can be easily. But naturally, you can pigment it. I mean, you can do it, you can increase the pigmentation. Of course, composition is also different, right? Two people, may have two different kind of, understanding. So like, let's take a chlamydomonas, okay? Hard-colored chlamydomonas, right? Or chlorella. And, you take a hematococcus. Hematococcus only accumulate astaxanthin, right? Because their expression, their genes, the pool of genes are more for the hematococcus. So the same gene is not in the chlamydia. So there was one, I think last class, Vijay, you presented, right? The chlamydomonas gene put into, the chlamydomonas, not chlamydomonas gene. Yeah, they put it in the chlamydomonas, they have expressed astaxanthin here. So, nanochlorops genes? Okay, so chlamydia nanochlorops. So, sir, especially those who are algal system, they live in different environment conditions. They have to adapt to that environment condition. That's why they evolved different pigmentation. But ultimately, in the beginning, every favorable condition, they will be happy to grow. But if you give something kind of light or nutrition deficiency, now they will be obviously they will have a different genes will, even though genes are there, their expression level will be a different. Let's say Ichiro Takashi has come, so he expressed differently. He was staying in Ella Hotel. He was not able to cross the road. We can cross, we can run, right? So we adapted to that environment. He could not cross, and he came back again hotel. I said it's not my cup of tea to walk on the street. Clear? Okay. Any other questions? Yes. No, this is a technique. Electron microscopy is a technique. So where when you go to Kelvin, low temperature, I mean they use a liquid nitrogen. So liquid nitrogen generally what it does, it's a freezing condition. When it is frozen the any kind of cells, not only the photosystem, you go with any cells isolated complexes, the reflection rate will be higher and the visibility will increase with the lasers. With the lasers, it will be like a more shiny. The visibility inside will be much better. So based on the section, you take a section and you freeze it and expose a laser, and you might see much better reflection. So whatever inside is there, that's what that nowadays that is the older, about 20 years back, 15 years back research, or 20 years back. So but now even much better, you can look at atomic level inside. Atomic structure is now determined by Pavitra is doing the same research what with the German collaboration. We are looking at atomic level, what at subunit level. That is the advantage with the technique. Yeah. Is it clear for you? Yeah.
There are two types of state transition. One is light induced and one is dark induced. Is there any difference between their mechanism?
That's what a mechanism is different. The common in both is light arresting complexes getting phosphorylated. That is the common. So here light is a signal, correct, inducing the state transitions in light induced state transition. In the dark induced state transitions, cyclic electron transport is circulating, where it is getting circulated by dark cycle, that is the metabolic cycle. Through metabolism, I think maybe I can show you the data. Oh yeah, here. So you can see here, through glycolysis, I didn't show you the glycolysis or metabolic pathway. From that, NADPH is getting, have you heard pentose phosphate pathway? Pentose phosphate pathway produces lot of NADPH, oxidized, when it get glucose is oxidized, you get NADPH. These NADPH is required, it goes, interact with PQ pool, then it will be circulated. Instead of coming like this through light, these NADPH are very good question. These NADPH are important in dark cycle. That is the signal.
Sir, this is a basic question, but I don't know the differences in between NADPH and
Okay, that is a metabolism. NADPH is, both are same. Here NADH, there is no phosphorylation.
NADPH, but when we are talking about glycolysis, then only we see the production of NADH. So I don't know.
Okay, NADPH is not a, NADPH is, it's a reduced, when PQ pool is, when the photosystems get, the final end products are NADPH and the ATP. So NADPH will not give any ATP molecules. It is a oxidant, it forms a redox oxidation mechanism, where they reduce, it requires NADPH for a Calvin-Benzil cycle to synthesize. So it will not go to mitochondria. So here, mitochondria produces lot of NADPH. When it breaks down glucose molecules, it produces energy compound, that is a NADH. This NADH is going to the respiratory cycle, where mitochondrial complex, where it generated NADH to ATP molecule. The ATP which we get in most of the animal system, or in the plant system, that is through NADH. NADPH is coming to synthesize, in the plant, the Calvin-Benzil cycle, it goes and interact with the CO2 and then goes to the Calvin-Benzil cycle to get the sucrose molecule. However, the NADH, in the animal system also have a NADPH without photosynthesis. That is through the pentose phosphate pathway, and that is not producing ATP. But NADPH is oxidant, is required for reducing the oxidation reaction. Anything like there is a stress mechanism, ROS produces. This goes and react with the glutathione S transferase. Glutathione is the one, it also antioxidants, where it will be playing, NADPH will be playing. NADPH will not, let's take example, NADPH will not produce any ATP, NADH produce ATP. Clear to you? All in chloroplast one. It's only chloroplast. Chloroplast again, chloroplast and thylakoid. Chloroplast is a big structure. I'm just globally I'm telling, and inside chloroplast, in the thylakoid it happens. Everything is in the thylakoid. But you would say the chloroplast. This is the chloroplast structure. I don't know. There is also the metabolic pathway is happening in the chloroplast. That's why I said chloroplast. Yes.
Sir, actually in winter season the plant does not have leaves, sir. So that time the in that they are using ATP, the life cycle will be going on actually. So ATP will be after the completion of ATP. I mean by using up the some metabolic activities occur in plants, sir. So how can they survive long days?
Yeah, that's what, it has a lot of, you know, if you are doing dieting, what happens? What happens to you? I'm not
Long day.
eating much. I'm not eating much, and you lose weight. Plants also lose weight. So because they metabolically they using carbohydrates or lipids, they get metabolized. That system is active. That means there is enough ATP is producing. And this is the example. The NADPH will be produced, and it goes to, this is the one is happening in the higher now the uh, state transition. This is the exact mechanism it applies to the in cold conditions. This metabolically they degrade metabolism, then through that NADPH comes and it operates, and ATP will be produced through the cyclic electron transport. The enough ATP there, they get synthesized. The dark cycle again will be there, Kelvin-Benson cycle through NADPH will be through the cyclic electron transport it happens. Clear? Not clear? Yeah, right. Thank you. Now